So, welcome back. Today, I'm going to be talking about cell transport. So, cell transport is just the movement of different substances in and out of cells. So, this movement is the way that cells maintain their homeostasis for the most part. Sometimes the principles of this movement make it so that the cells actually lose their homeostasis. Right? The cells only have certain amount of regulation over these mechanisms. And so, depending on the conditions of the overall body, sometimes those conditions force the cells into states that they are not comfortable in and that they do not perform ideally in. Right? An instance of that would be if you have drinking too much salt water. And so when you drink too much salt water, the water inside of your cells, which is nice and pure and clean, actually leaves your cells and goes into your blood. Right? And so your cells now are low on water, which is obviously bad, right? And dehydrated. So that's just a very quick example of how sometimes these things aren't actually regulating homeostasis under the wrong conditions. But in general, this is how cells maintain the levels of the chemicals and stuff that are inside of them. There are two main categories of cell transport. There is passive transport and there is active transport. So we're going to look at passive transport first. The defining feature of passive transport is that it uses no ATP. There's no energy needed. It happens through the natural progress of diffusion. And diffusion is the tendency of substances to move from an area of high concentration to an area of low concentration. And so the way that I usually like to demonstrate this is I'd like you to imagine that somebody is at a opposite end of a room from you. Okay, imagine a classroom-sized room, and they're sitting in the corner, and they got this little tinfoil wrapped thing, and you don't know what's in it. Right? You're just sitting there and you're watching. You're like, what are they opening? Why are they doing that in the classroom? What are they doing? Right? So you're watching them, and they peel it open, and you still can't see what it is. Right? And they're looking at it like it's just the most delicious little thing in the whole wide world. It's a cute little snack they're about to eat. Right? And all of a sudden, your side of the room starts smelling like tuna fish. And you're like, how does this side of the room smell like tuna fish? And you're like, oh, they must have a tuna sandwich, right? And so the tuna molecules were highly concentrated inside of their little tinfoil package, right? When they open the tinfoil package, those molecules will spread out the room to the area where there was low concentrations of tuna molecules, i.e. my nose. Right? And so the tuna molecules floated over, got in my nose, made me all upset because I hate the smell of tuna. And I said, why are you eating tuna in my classroom? That's diffusion. Okay? Molecules move from high concentration, where there's a lot of them, to an area of low concentration, where there's less of them. Okay? Happens all on its own accord. Now, the thing we have to keep in mind is that concentration is not a measure of the absolute amount of the molecule in the area. It's a measure of how much of that molecule there is compared to all of the molecules in the space. Right? So in my example, I was looking at only tuna molecules. And if there's only tuna molecules, then the area with 10 tuna molecules has a higher concentration than the area with 2 tuna molecules. Right? There's more over there, they're going to move this way. Okay. The reality is it's not just tuna molecules, right? There are oxygen molecules and carbon dioxide molecules and sulfur molecules and methane molecules and all those human smell molecules that fill up the classroom way too quickly. Ooh, human smells. Right? And so we have to compare how much tuna molecules there are to how many total molecules there are, and that gives us our concentration. And that's just a simple ratio. Right? And so here in my whiteboard model, I have circles and I have squares. On the left side of the model, we have 10 circles and 4 squares. That gives us a total of 14 molecules on the left side of my barrier, which is represented by the dotted line. Again, 10 of those are circles. So we take the 10 circles, we divide it by the total of 14 molecules altogether, and that gives us our concentration ratio for our concentration of circles. In this case, it's 10 over 14, which can be simplified down into 5 over 7. Okay. On the right side of our barrier, okay, we have 3 circles and 4 squares. That gives us a total of 7 molecules altogether on this side, 3 of which were circles. So our circle concentration is 3 over 7. We compare the two numbers. So I got 5 over 7 and I got 3 over 7. Right? 5 over 7 is bigger than 3 over 7. So it's going to move from the big side to the small side. Okay? So my circles are going to go that away. Okay? Net movement going to go that way. Okay? And that's how diffusion works. So passive transport is driven by this principle of molecules' natural tendency to move from a high concentration to a low concentration. Okay? And this can be into the cell or out of the cell, doesn't matter, goes both ways. Okay? We phrase it as going down the concentration gradient or along the concentration gradient. Those are both terms for the natural tendency of molecules to move with the concentration gradient. The words that I've squared off in blue throughout this presentation are vocabulary words, just things you should pay special attention to knowing as words themselves. Okay. Now, as opposed to passive transport, active transport requires the input of energy to move the molecule. And the reason it needs energy is because it's going to go the opposite direction from that natural inclination. Okay? This is going to move the molecule up or against the concentration gradient. Okay? So we're going to go from a low concentration gradient to a high concentration gradient. Okay? And so if you think about going from a low place to a high place, you got to go uphill. It takes more energy, right? Same idea, right? So if you're going low to high, requires energy. You go high to low, doesn't require energy. Okay? So active transport always requires energy, and it's typically moving something from a low concentration to the high concentration, opposite the usual. Okay. So here we have a model. Now you see here, I've changed my barrier representation here because I don't want these things to pass easily, right? If these were allowed to just pass through this barrier without any restriction, they're going to follow the concentration gradient, and that's not what I want in this example. Okay? So now here, these circles can't just go through this barrier. There's no space for them. They have to pass through this little thing that I've drawn here. Okay. This is representing a protein channel, which we'll see in the next segment of this presentation. Okay. But for now, it's just think of it as a doorway. You know, it's like a spinny door that you can walk through one way, but you can't walk through the other. Okay. So this spinny door only moves from the right side to the left side. Okay. That's the only way that the spinny door will move. But the spinny door is going to require energy to move it. Okay. And now this spinny door is going to take my circles and it's going to push my circles from this side to this side. That's what's happening in this model. Okay. If we look at our concentration levels though, right, I have a concentration of circles over here, which is 5 out of 7, because I'm back to that 10 out of 14 that I used above. Okay. And over here I have a concentration of 2 out of 7. Okay. So I'm trying to move my circles against the concentration gradient. I'm trying to go uphill. And so I need that energy to push the molecule uphill, basically. So the circle is going to engage with this protein channel. We're going to throw some energy at it in the form of ATP. The protein channel is going to use that energy to push the circle uphill against the concentration gradient and say, bye, circle, get out of my life. Okay? So that's active transport. Again, needs energy. That's what makes it active. Okay? So that's passive versus active. And the last topic that I'm going to look at here in this introductory video is the selective permeability of the cell membrane itself. Okay? And that's just a fancy way of saying that not everything can go in and out of the cell. Right? There are restrictions about what kind of molecules can enter or leave the cell without issues. The cells are very physical things, right? The membranes are very physical things. The molecules moving across them are very physical things. Okay? And if you try to take a big thing and put it through a small hole, I'm sure you know it doesn't work, right? And so the molecules that are moving in and out of the cell across the membrane are going through holes. That's the only way to get through something, right? And so the way the cell membrane is structured, there are little holes in them. There's little gaps between the pieces that build up the membrane. Molecules that are small enough to fit into these gaps can just move along with the concentration gradient, right, through diffusion. So if you have this tiny little molecule and it can just go straight through the membrane, it's going to go from the high concentration to the low concentration all the time. That's the only option it has. Okay? It will always follow diffusion if it's small enough to go through the membrane. Okay? The membrane also has another property where it has a very thick region in the middle that is nonpolar. Right? And so polar substances get rejected by that middle region. So you have to be small and nonpolar to go straight through the membrane. Larger molecules or polar molecules or charged molecules such as ions, right? Like sodium ion, which is a cation. Is it? Yeah, no, yes. Sodium is a cation, which means it has a positive charge, right? And its complementary ion is the chloride ion, which is an anion, it has a negative charge. Okay, so those kind of things can't go through the membrane on their own. Okay, if it's too big, if it's polar, or if it's ionic, it can't go through the membrane. Has to go through a special little gateway. Okay. So here I have my very rough illustration of what the cell membrane is kind of designed like. Okay. So here's my cell. Okay, now remember the cell's three-dimensional, right? It's a sphere, it's not really a circle. Okay, so this membrane is going all the way around the cell in all dimensions. Okay, it's around the whole thing. So I've just zoomed in on this very tiny segment of the cell membrane, and it's in only two dimensions. Okay. The cell membrane is made up of mostly these structures, which are a phospholipid. Okay, this circle part is representing a phosphate group, where there's a phosphorus surrounded by a bunch of oxygens. Okay, and the squiggly lines are representing the lipid part of this. These are lipids, right, which we learned about in the macromolecule section. Right? So these are just lipids. Okay. Now the cell membrane is a phospholipid bilayer, bi meaning two, because it has a double layer of phospholipids. It's very well named. Somebody did a good job that day. Okay? And the lipid tails touch each other, and the phosphate heads look into the outside of the world and the inside of the cell. Okay? The phosphate heads are what's called hydrophobic. No, Mr. Lavender, that's dumb. Hydrophilic. Hydrophilic. I'm going to write that, especially since I said it wrong the first time. Right, so phosphoheads, hydrophilic. Okay. Derived from the suffix phile, right? Phile, which means one who loves, right? Like a bibliophile is one who loves books. Okay. Hydro meaning water. So these are water-loving things. So the phosphoheads are water-loving. Okay. The tails, the lipid tails, right? They're lipids, so they hate water. They absolutely hate water. They're like, get away from me, water. You're stupid. And they are hydrophobic. Okay. Like phobia. So this suffix is related to phobia. They're afraid of water. Okay? And so obviously inside the cell is watery, outside of the cell is watery. Okay? And this layer keeps the two watery sides from crossing into each other. Okay. So because the inner layer is nonpolar and hydrophobic, okay, it's nice and thick. Like this is the longest part of the membrane, right? This is the thick part. Anything that is the same as that can pass through because they're friends. They're like, this is cool. But the ones that are different than this is it rejects them. It says, get out of here. Right. So this nonpolar region is cool with nonpolar molecules. So if a molecule is small, it's small enough to fit between the space between the phospholipids, right? There's literally spaces there. If it's small enough to fit in that gap, and it's non-polar enough to hang out with this thick section here, it just goes right through. Foop, no problem. Sneaky Pete. Okay? However, if a molecule is bigger than the gaps, or if the molecule is polar or ionic, it has to go through these protein channels, which in the simplest form, just tubes of proteins that things can pass through, right? They do get much more complex. Some of the protein channels are shaped in very specific ways to only allow very specific molecules through. Some are shaped even more specifically to only allow specific molecules through under specific conditions, right? So it does get way more complex than just being a tube, but for the purposes here, just think of it as a tube that these molecules can go through. And it's made out of proteins. And so, yeah, that's what I wanted to introduce to you before you got into the next class. And hopefully that gave you a good enough base where when we approach the pole that we're going to look at, which is about this membrane stuff, we'll look at that in more detail, that you'll be all set. Okay? So we have two types of cell transport. We have passive, which uses diffusion to move molecules from a high concentration to a low concentration. We have active, which uses energy to push molecules from a low concentration to a high concentration. And all of the underlying principles that are controlling these mechanisms come down to the selective permeability of the membrane, which is derived from the structure of the phospholipids themselves, determining that small nonpolar molecules can just slip right in for passive transport very easily, whereas larger or polar or ionic molecules have to utilize the protein channels to move from one side to the other. Okay? And that's it. Hope you learned lots. Have an awesome afternoon or morning or evening or late night, whenever you watch this. Just have a great time. Bye.